November 19, variant 3. Photosynthesis occurs in chloroplasts. A chloroplast suspension can be made by extracting the chloroplast from green leaves. Okay, he will extract the chloroplast by extracting it from, he will create a suspension by extracting the chloroplast from green leaves. Okay, what is he going to do? Blue dye DCPIP is used to investigate photosynthesis. So the results here, he will measure the results using the blue dye DCPIP. This is the results he is measuring. During photosynthesis, how is he going to measure the results? During photosynthesis, the blue color of the DCPIP changes and eventually disappears. So if photosynthesis occurs, the blue color will disappear and it will become colorless. The remaining mixture appears green due to the suspension, the chloroplast suspension. يبقى ال DCPIP will change from blue to colorless. When it is colorless, the color of the chloroplast can be seen. You can see a green color. ده معناه إن ال photosynthesis happened. This means that photosynthesis has occurred. Okay, so this is the results you are going to measure. DCPIP, when it changes, green color will appear. This means photosynthesis occurred. طيب, a student investigated the effect of light on photosynthesis. I want to investigate the effect of light. But what is the change variable here? The change variable will be the light. This is the change variable. I will change the light and see what happens to the photosynthesis. The student crushed 8 green leaves in an ice-cold buffer solution. The buffer solution is a solution that adjusts the pH. The mixture of the crushed leaves and the buffer solution was filtered to form the chloroplast suspension to get an extract of the chloroplast. The student was given 2 test tubes. One was wrapped in aluminum foil to exclude the light. The second was not covered so that the light could reach. يبقى one of them, the light is present, the other, the light is absent. A plastic pipette was used to add 2 cubic centimeter of chloroplast suspension to each of the 2 test tubes. I added the same volume of the suspension. يبقى this is a controlled variable. Plastic pipette, we know that the pipette is not our best option because the volume might be a bit different. Okay, so this is an error. A syringe was used to add 5 cubic centimeter of DCPIP to each test tube, same volume for each, but this is a controlled variable, constant. A stopper was placed. Both test tubes were placed in a water bath containing ice-cold water near the bright light. Is it going to remain at the same temperature? No. Whatever the temperature is, cold, hot, whatever the temperature, if you left the water bath, it will change temperature. So this is a source of error. It has to be a thermostatically controlled water bath. Water bath was positioned near bright light. The test tubes were left for 5 minutes. You left them for the same time. But this is a controlled variable. After 5 minutes, the student observed and noted the color of the liquid. يبقى anything with color, بقول له, I have to say that no, this is not accurate. This is a source of error because it is difficult to judge by the eye. It is better to use a colorimeter. Okay. The test tube that was not wrapped in foil, they removed the foil from the cover and observed the color in the other test tube. Type, what happened? The test tube in the light gave me green color. Why did it give me green color? Because the DCPIP changed from blue to colorless since in the photosynthesis occurs in the light. So the green color of the chloroplasts can be seen. While the one in the dark, the DCPIP remained blue because the photosynthesis did not happen. Prepare a table to record your results. As we always say, the changed variable is on the left. A presence of light and dark on the left. Results is on the right. The color of the DCPIP, and you will write the results. The one here, the color of the solution here was green, okay, while here it was blue. This is the table that should be in your paper. What was the variable changed? Independent. And what was the dependent? Independent was the light. This is what I changed. The dependent was the color of the solution. Color of the solution. If it changed colorless, the green color will appear. Okay? So what were the variables kept constant? Anything. Variables that were kept constant. I can say the volume of the chloroplast suspension, volume of DCPIP, anything that I already marked. Volume of chloroplast suspension, volume of DCPIP. What is the conclusion for the results? The conclusion is that photosynthesis needs light to occur. Photosynthesis does not happen in the dark. Photosynthesis needs light to occur. In step 6, both were placed in a water bath containing ice cold water for 5 minutes. The thermometer was placed in the water bath. As we said, definitely the water bath left on its own كده, the temperature will change according to the room temperature. What was the temperature at the start? It was 0.123 degrees Celsius. Include the unit in order not to lose a mark. 3 degrees Celsius. Temperature after 5 minutes, it was 8 degrees Celsius. Okay, the investigation was not repeated. Why do we repeat? What is the reason for the repeats? We always write 2 points. Actually, 3 points. To calculate an average, okay? To identify anomalous results, and both of these points will increase reliability. To increase reliability of the experiment. We repeat the experiment to make sure that the numbers you get is correct. There is no anomalous results. Calculate a proper average to increase the reliability of the experiment. Okay. Identify one other source of error other than the repeat, and suggest an improvement. We already said a few errors. We can say that pipettes are used. Pipettes do not give accurate volume, okay? So use a syringe instead or a burette. Temperature is not maintained. As we said, the temperature will change to the room temperature. Temperature is not maintained. How do we improve it? We use a thermostatically controlled water bath. Use thermostatically controlled water bath. I can also say that it is difficult to judge the end point of the color. You cannot judge exactly when did it change the extent of the color, when is it colorless. So it is best to use a colorimeter. Any of the errors. Okay, a student wanted to investigate the effect of temperature. In the above experiment, he was investigating the effect of light. Here, I want to investigate the effect of temperature. يبقى this is my changed variable. This is what I will change. How are you going to see the results? He asked you to use DCPIP the same way as he did. I will follow my steps. The first step is replica. How many test tubes are you going to use? Okay. Use 5 test tubes. طب what are you going to put in the test tubes? The chloroplast suspension. Chloroplast suspension, just like he did. Crush 8 green leaves in water, okay? And then filter to get the chloroplast suspension. Same as he did. Crush 8 green leaves with water, then filter to obtain suspension. What am I going to change? I'm going to change the temperature. Place each test tube in a water bath with different temperature. This is my independent variable. And then you can give him examples of the temperature given: 20, 30, 40, 50, 60 degrees Celsius. What are your controlled variables? You are going to change the temperature. What has to be the same? Put same volume of chloroplast suspension. Same volume of DCPIP. Put all in the same light intensity. Careful that in the above experiment, he changed the light. For me, I'm changing the temperature. So it has to be placed in the same light intensity. Okay? What am I going to measure? How am I going to measure the results? You will measure the time. I can measure the color. I can observe the color. Okay? But here I have several test tubes and I want to see which one changes faster. So, how am I going to measure my results? You will measure the time taken for it to change to colorless, for the DCPIP to become colorless. How do I know that it became colorless? You will see the green color of the chloroplasts. Measure the time taken for the DCPIP to become colorless. This is your dependent variable. And then at the end, you can write an improvement. Here we are using different water baths. One will be 20, one will be 30, but each of them has to be maintained. Each of them, the temperature has to remain as it is. The 20 has to stay at 20, 30 has to stay at 30. So how do I say it? Maintain temperatures using thermostatically controlled water bath. Safety precaution, wear goggles, and then repeat experiment and calculate an average. Repeat experiment at least 3 times and calculate an average to be more reliable. Two, photomicrograph part of the lower epidermis. Make a large drawing. You need to take care. He gave you the lower epidermis. This is 2 guard cells. This is 1 guard cell on top. Oh, this is 1 guard cell. And this is the other guard cell. And the opening in the middle is the stoma. Here is a lower epidermal cell, and here is another lower epidermal cell. The bottom one is larger than the top one, so you have to draw it like this. Also, whenever you see a very dark layer, this means that this layer is thick. So you will draw it as double lines, but please, no coloring, no shading, just lines. So if I'm going to draw this, I will draw one guard cell and the stoma in the middle, the second guard cell, lower epidermal cell here, and a bigger one at the bottom. The inner wall is thick, so I will draw it as double lines. This is my drawing. This is what I want to see. Guard cell, epidermal cell. The bottom one is bigger, and thickening of the inner walls of the guard cells because they were dark in the photo. Okay. Measure the length of the line MN as we are used to every time after the photograph, after the drawing, there is a magnification question. Measure the line MN by your ruler. It will be 3.1 centimeter, but here he gave you the millimeter. So I will multiply by 10. It will be 31 millimeter. Calculate the actual width. Put your triangle. Magnification equals image over actual. I want the actual, then it is image over magnification under the picture. 400. It will give me a number. I will round up to 2 decimal places. Round up to 2 decimal places. It will be at the end 0.08. Okay. A student investigated the effect of sucrose on the stomatal opening. I'm investigating the effect of sucrose concentration. But this is what I changed here. Independent variable. This is the independent variable. Strips of epidermis from the leaves were placed into different concentrations of sucrose solution for an hour. We measured the width of 10 stomatal openings from the epidermis in each sucrose solution. Circle one measurement that could be anomalous. طيب, let's check the numbers here. 5, 6, 7, 5, 5, 6, 7, 5, 7, 6. Okay. 3, 4, 3, 3, 4, 3, 2, 3, 3, 2. Okay. 1, 2, 1, 2, 1, 1, 5, 1, 2, 1. This is a wrong answer. This is a wrong measurement. Why? It is anomalous because if he asks me why is it anomalous, I will say because there is a big difference between it and the other numbers. Big difference between this number and the other numbers. Calculate the average. You will add all these numbers over 10. The total, the sum over their number, it will be 3 micrometers. I will add all these numbers, sum of all the numbers over the numbers. All of these over 10. Pineapple plants are adapted to grow in hot, dry conditions. We investigated the number of stomata opening in the pineapple plants at different times of the day. Different times of the day. Okay. Calculate the percentage change. Between 16 and 20. Between 16 and 20. The rule is final minus original over original times 100. So it is 51 minus 10 over 10 times 100. Calculate it on the calculator. It will be 410%. And then, of course, he will ask you to plot a graph. Here, it was a bar chart. Bar chart. Left side of the table is always on the x-axis. Right side is always on the y-axis. I will draw bars. The name of each bar will be the timing. I put a scale on the y-axis. You do not need a scale on the x-axis. البars have the names. The rule is the bars should be equal in width and the spaces between them should be equal. Each of them will have a name underneath. The average number of stomata, biggest number was 77. You have 8 boxes, so my interval was 0, 10, 20, 30, 40, up till 80. I wrote the labels, time of the day, average number of stomata, drew a bar for each timing, and wrote the timing at the bottom. Describe using your graph how does the stomata changes throughout the day. Using my bar chart, as I can see, the average number of stomata opening decreases until 12 and then increases. Highest number was at 0.00 and then it decreased till 1200, increased again till 800. I can use numbers. It was مثلا 50 something, 51 at 800. So you will just describe that data in front of you. He just needed 2 marks here. If he needs a 3rd mark, I can use data code. I can use numbers. Okay. It was how many was there at 4 مثلا and how many was there at 8. When the fruit ripens, starch contained is converted into sugar. What is the name of the substance used to test for the presence of sugar? Iodine solution. How can you safely test for reducing sugar? When he says safely, you have to write a safety precaution. What is the procedure? Add a sample of juice, then add Benedict reagent. After adding Benedict, there is an extra step. Heat in water bath. If there is sugar, it will change to orange red. What is the safety precaution? Either you can say wear goggles to avoid splashing of the reagent to your eyes, or you can say use a test tube holder in order to avoid burning your hands because of the hot water in the water bath.